If you've listened to Brewlosophy at all, you know how much we love Imperial Yeast. The company was founded on the ideal that if you're going to do something, do it right. From their Imperial Pitch-Right pouches, packing a whopping 200 billion cells of healthy and viable yeast in each pouch, to their commitment to commercial customers guaranteeing 10 of their most popular strains are in stock for orders up to 20 liters, or the shipping is free, Imperial Yeast does it right. No more worrying about whether you need to make a starter or propagate your yeast prior to pitching. Imperial Yeast makes it easy for the home brewer and commercial brewer to obtain direct pitches at proper rates when you need it. To place a commercial order or to get more information about everything that Imperial Yeast has to offer, head on over to imperialyeast.com. Welcome to the Brew Lab. Most commercial brewers have some form of yeast repitching scheme whereby yeast from the last batch of beer is used to ferment the next batch. Intermediate and advanced homebrewers often do the same thing, harvesting yeast from prior batches for later use. But how many times can you reuse the yeast? And the subject of this episode, how does the yeast change over time as you repitch? I'm your host, Cade Job, and today in the lab I'm speaking with Dr. Riddhiman Garge, post-doctoral researcher at the University of Washington in the Department of Genome Sciences. Riddhiman's research up to this point with yeast has focused on the role of yeast as a model eukaryotic organism. That is, studying brewer's yeast could tell us a lot about how other eukaryotic cells, like human cells, for instance, might behave under evolutionary pressure. Essentially, how might human cells change over time? And we can answer those questions by potentially looking at brewer's yeast. Well, with brewer's yeast, we're forcing rapid changes to the yeast genome in almost every fermentation. In fact, there's an incredible amount of evolutionary pressure on yeast. Think conditions without oxygen, fermenting at cold temperatures, fermenting at hot temperatures, fermenting quickly in the presence of a huge amount of sugar, and then alcohol and CO2. Studying how yeast changes over time can tell us a lot about how other cells might behave in similar conditions. And not to digress too far into human health topics, this research could actually help us understand where human cells might go in the future under the right conditions. Okay, but I'm not a medical doctor. I'm a brewer, and as Riddhiman found out, brewers love brewer's yeast and want to know how it's changing over time. Why can I only repitch my yeast for 10 to 20 fermentations before I have to go throw it out? Why does my beer taste better after the third repitch cycle than fresh yeast? These are all anecdotal questions that I hear brewers ask, and they're all interesting things for researchers to look into. Researchers like Riddhiman and like Mariana Barreto Carvajal Pinto, who looked at proteins to see how the beer matrix is changing. So in the case of Mariana, she looked at how bitter acids and proteins are interacting during the boil. Well, Riddhiman looked at how proteins change over time due to serial repitching. He used the popular Weinsteffen Hefe yeast, Imperial's G01 Stefan, and the results were really interesting. I think you'll be amazed at how far yeast can change in even just a few repitches. We'll get to all that and more right after the first break. Now, we've wrapped up 2023 and we're on to 2024, and as we look at the year that's passed and look forward to the new year, I'm thankful, of course, for all of our listeners and especially for our patrons. We wouldn't be able to do this thing without you, and if you're not currently a patron, I encourage you to head over to patreon.com/Brewlosophy and check out the monthly contribution levels. By becoming a patron, not only do you help us keep this going, you also get access to Brewlosophy contributor recipes that we've never published, new discounts each month to YakimaValleyHops.com, and for $3, access to a monthly live Q&A session with a special guest from the brewing industry. We've had all kinds of guests like John Palmer, Jamil Zainashev, Vinny Chilurzo, even Mr. Brewlosopher himself, Marshall Schott. And when you sign up, all of these prior sessions become available to you on our private Patreon Facebook page. So please head over to patreon.com/Brewlosophy to find all the information you need to get access to this awesome resource. And thank you to everyone that's left a rating or review of the show. If you haven't yet left a rating or review, I'd love for you to do so. Every rating helps us improve the show, and it helps others like you find this show. I read all of the reviews, and I love hearing from you. If you haven't yet had a chance to leave a rating or review, please go ahead, take a moment to help us out. Feedback is brought to you by the clever folks from North America's leading hop supplier, Haas, who developed Incognito, a 100% all-natural hop product crafted to deliver highly concentrated flavor while maximizing brewing efficiency and reducing process loss. You wanted more varieties, and the Haas team heard you. Incognito is now available in 8 of the most popular Whirlpool hop varieties: the Centennial, Chinook, Citra, Equinox, Eldorado, HBC 472, Mosaic, and Sabro. All the hoppy goodness you expect without adding any actual hop matter. means less process lost, increased profits, and best of all, more beer. You can learn more about Incognito and all the other innovative products Haas has to offer at johnihaas.com. That's John, the letter I, HAAS.com. All right, listener John from Sioux Falls wrote in a few months ago, and he said, "Hey, Cade, I love the Brew Lab. I've been slowly catching up on missed episodes, but I'm really digging the science and answering questions I've had about brewing. To that end, I've started harvesting and repitching yeast from batch to batch with varying degrees of success. Could you do a show about the science of repitching yeast? Thanks for all you do." So, John, yes, of course, here it is. It took me a few months, but this is the first of what I hope will be at least a couple of episodes on this topic. This first one, of course, about how yeast proteins change over time with Dr. Riddhiman Garge, but then also a follow-up with Dr. Maitreya Dunham about serial repitching, how many generations is too much, and what changes we see in terms of outcomes to brewers resulting from serial repitching. So thanks for writing in with the show idea. I'm glad that I'm able to accommodate and have this show, and I hope that you enjoy it. All right, I'm going to take a quick break, and I'll be back in a few minutes with Dr. Riddhiman Garge talking about how yeast proteins change with serial repitching. The dry yeast revolution is here, and Cellar Science is leading the charge. Cellar Science has expanded the world of dry yeast far beyond the drab landscape of yesteryear. With over 20 strains of beer and wine yeast now available, you can brew any style. Not to mention that their unique growth and harvesting process greatly increases lipid levels in yeast cell walls, removing the need to aerate your wort on the first pitch and allowing you to directly pitch without rehydration. Couple that with the other advantages of dry yeast, such as higher cell count and viability, and you can clearly see why commercial breweries across the U.S. are using dry yeast and choosing Cellar Science. On its surface, fermentation sounds easy. Pitch yeast into sugary wort and make beer. But the brewing process and the mechanics of what actually happens during fermentation are anything but simple. Yeast is a widely studied organism, as we can see from the dozens of episodes of this podcast on yeast, but there are still things we don't know, like how does the yeast change during fermentation? How does the fermentation itself change after serially repitching the yeast? Well, my guest today takes a proteomic look at answering these questions, and that guest is, of course, Dr. Riddhiman Garge of the University of Washington. Riddhiman, welcome to the Brew Lab.
Hey, Cade. Thank you so much for having me. Pleasure to be here.
Oh, I'm glad to have you. Riddhiman and I actually met face-to-face at the MBAA national meeting, which happened earlier this year in 2023. He gave a great talk on yeast genetics and what happens to the yeast genome during fermentation. And it was a really, really great talk. And so I talked to him and said, hey, we should do this and we should get you on the podcast. Now, like I just mentioned, I mean, brewer's yeast is one of the most studied organisms on the planet, and yet you still found a new way to study it. That's pretty cool.
Yeah, this is really exciting because, this project has been kind of a labor of love for me in some ways, but more so, I had been exposed to baker's yeast, early as an undergrad, studying proteins that form channels to transport molecules like sugars and into yeast cells. And during my PhD, I used it as a tool to build and study human systems. And often that's quite powerful to get at understanding the implication of these systems in health and disease. Yeah, so this was really one of the side projects in my lab as I was wrapping up my PhD. And to answer your question of like how I started working on this project, it was really kind of serendipity. And that came out of me crossing path with these two really amazing people. The first one was Chip McElroy from Live Oak Brewing. He's the founder. And Chip himself was trained as a biochemist and visited the department at UT Austin, where I got my PhD, and to talk about what you can do with your PhD on careers outside of academia to impressionable grad students such as myself. And especially how you can use your skills in things that go beyond the lab. And so with extensive interactions with Chip and his Live Oak brewers, what kind of stood out very obviously was that Chip and the brewers were like really curious to understand how fermentation, how yeast behave during fermentation and how the environment impacted yeast in terms of how the beers they produce and the properties they impart to it. And I think the second person that was really key in terms of getting this project off the ground and getting it to where it was today was Maitreya Dunham, who is a professor at the Genome Sciences department here, where I work currently now at the University of Washington. And I was fortunate enough to collaborate with her on this project. And her lab actually doesn't use proteins, but they study genes, and more so genomes, and how genomes evolve and adapt. Over the course of evolution, using brewer's yeast again as an experimental tool. And so our paths crossed when she visited Austin for a sabbatical, working on understanding how the genomes of specifically brewing yeasts adapt to fermentation environments. And then, as I was finishing up grad school, that's when I realized that we could really leverage my old lab's expertise at UT Austin, which is taking these kind of systems-wide views, particularly profiling proteins from the insides of cells, with the Dunham lab, where they study adaptations to industrial environments. And Live Oak was like the perfect test case, because they were also really interested in understanding how yeast strains change over the course of fermentation, particularly at the molecular level.
I love it. I mean, this is one of the things. This is one of the reasons why I wanted to start this podcast, was because brewing science isn't necessarily always just about brewing, right? I mean, this is like we look at like the discovery of the yeast cells. Sure, Louis Pasteur was looking at beer when he found the yeast cell, and he was doing brewing science, and we discovered that microbes exist, right? I mean, all these different adaptations and things that we've studied over time have been extensions of brewing science. And I love it. And I love whenever, two people can come together over a beer, or three in this case, right? Three people come together over a beer and come up with an amazing research project and a new way to look at brewer's yeast, which is, of course, near and dear to the heart of all of the listeners that are listening to this podcast. Now, just a little bit of introduction for you. So you are a postdoctoral researcher at the University of Washington in the Department of Genome Sciences. And as you mentioned, recently got your PhD in biochemistry from the University of Texas at Austin. Hook 'em horns, by the way. I am originally from Austin. And by the way, I love Live Oak's brewery. That Hefeweizen that they have, as well as they had a smoked Grodziskie on there for a while, which was a sour smoked beer. Those were fantastic beers. I love those beers and love that brewery. It's a fantastic place. Of listeners, if you're ever out there in Austin, go check it out. And so I would normally ask, how did you become interested in studying, the brewing yeast? But I think you just answered it. So it sounds like you guys were having this conversation, and this study just sort of naturally evolved from each of your passions.
Yeah, no, that's absolutely true. I mean, like you're a fan of the Live Oak Hefeweizen. That's one of my favorite beers too. And in the Pacific Northwest, we are not afforded the luxuries of lagers and pilsners the way we are, and where we were in Texas, as you know. But essentially, no. Talking to Chip is like one of these really deep thinkers of like what molecular things contribute to the flavor, and I think he's also a real champion for community building and trying to bring people from different disciplines together. And I think if not for that kind of drive, I don't think we'd be able to do this project. And I think to answer your question, it was natural, but it is also just like happened to be that we ended up going to Live Oak often enough that they were really nice about letting me go into their brewery and work with them, and kept letting me come back. So it's very natural, and it's really complemented my graduate work, which was mainly using yeast to answer these questions about how genes and systems evolve. But now this is looking at it through an industrial lens.
Yeah, exactly. And so that's, of course, what you were looking at in this study that we're going to talk about today, is looking at how Saccharomyces cerevisiae, so that is brewer's yeast, how it changes through fermentation. So from the part where you pitch it into the yeast through fermentation, and then again, after it's been serially repitched, and looking at all this through a genomic and as well as, in your case, a proteomics lens as well. What's happening with the protein proteins, because as we've learned recently on that episode with Glenn Fox talking about free amino nitrogen, we know that proteins and nitrogen are incredibly important for yeast. And so this was really cool. So just really briefly, I mean, you analyzed the genome of a Hefeweizen yeast and then looked at the proteins that were being produced by that Hefeweizen yeast. I'm guessing that was the Hefeweizen yeast that they use in live oak hef. And looking at it again after 14 serial repitchings. So I do want to make a note here too that Dr. Maitreya Dunham is going to come on the show in a few episodes in the future to talk about yeast genomic changes after serial repitching. We'll touch on that a little bit here because it's relevant, but the main focus of today is going to be to talk about what proteins change and also why those proteins might be important to brewers. So we got to start at the top then. So we often say that brewers make wort and yeast makes beer, and of course that phrase sounds really simple. But how complicated is fermentation?
Oh, I mean, when I started this project, I literally had the same view as you, right? Like basically,
yeah.
And I'm no brewer by any means, and I probably will never be a brewer as amazing as what everyone else out there is doing. But basically, I thought it was taking wort, which is rich in sugars and nutrients that the yeast lives off of. You feed it to yeast, and it uses its enzymes and its proteins to convert that sugar into alcohols, right? So, that's kind of the summary of, your one sentence summary of like fermentation, right? But obviously, what we noticed is that that's not the case, because if that's really that straightforward, then all beers would taste the same. All yeast would make the same types of beer, but obviously, that's not what we see. Not only do strains make different styles of beer, but the molecular mechanisms that underlie beer flavors, the compounds that they produce over the course of fermentation are different between strains. And this is one of the central motivations of the project is like, before we can really understand how different strains produce their own characteristic flavors and beer styles, we need to map the molecular changes across time in a single yeast strain, and so that we can make a map. And then after that, we can go and look deeply in that map and say, hey, this protein is doing this function at this time, and then that's the reason why you're making this particular flavor compound. And so that's where we want to get to. And so this is our project is kind of the first step at getting towards those questions.
Oh, wow. That's a really amazing, that'd be an amazing tool if we had that for brewers, where we're able to look at like, okay, this is the proteins that your yeast strain is making, and this is how it's going to impact your beer. And these are the actual, chemical differences and molecular differences between the yeast strains. And again, talking about that, I totally agree with you, right? This is sort of how I always approached beer too, is like, okay, just pitch some yeast and then you're going to, it's going to turn the sugars into alcohol and CO2. And that's the main point. And it is still the main point, right? We want alcohol and CO2 in beer. But as you mentioned, different yeast make different beers. And there's a whole range of things that may influence that. And you mentioned this in the paper. Other people have looked at, different aspects of this, right? Like they've looked at esters, or they've looked at higher alcohols, or, other pieces of this. But your paper was the first that looked at this from a protein standpoint. And one of the things that you mentioned was you talk about that yeast faces challenges as soon as it's introduced into sugary wort, and it has to address those challenges in order to survive. So is that where proteins really sort of play a major role, is in how the yeast, essentially survives in sugar wort?
Oh, absolutely. I think, previous studies have looked at RNA and genomes and genes, and how strains are different because, certain strains lack certain genes, and the expression, or like the way that certain genes get used in different strains are different. But proteins are actually the functional units of the cell, and they're actually involved in nearly every cellular process, from cell division to alcohol production to responding to the environment and stresses. So I think looking at the protein level really gives you you're looking at it at the functional level at which, proteins are the effectors of the genes, basically. So the genes have the sequences that make up the proteins, and proteins actually execute the tasks. So looking at it at the proteome tells you what that uh, cell is doing at that particular time in response to its environment or during fermentation.
Oh man, yeah, and that's really cool. And I can't wait to dive into some of those results too. Like, what is the yeast doing at the beginning of fermentation versus the end of fermentation, and how does that change when the yeast is repitched? And as an example, you just mentioned that the proteins are sort of the functional aspect of the cell. They're the ones like that are doing the things, right? The cell is converting sugar into alcohol and CO2, but it actually, it's not like that just magically happens, right? The cell is creating proteins and enzymes to do those different tasks. The sugars have to get inside the cell, right? And then they've got to get broken down and then turned into, again, I'm not going to go through all the molecular processes here, but turned into acid aldehyde, and acid aldehyde gets turned into ethanol, right? That's the sort of, simple process that makes alcohol. But again, the whole point of this is like sugars create stress on the cell, right? Like, and ethanol too, like these stresses are like, they're sugar outside. So the cell wants to, either bring in sugars or adapt to its surroundings. So it needs to beef up its cell wall and things like that. So can you talk to some examples about like what proteins are doing, like from a functional level?
Yeah, so I mean, that's absolutely true. So I mean, proteins are the things that take the sugars from the outside of the cell to the inside, and they are the ones that convert the acetaldehyde into alcohol, all of that. But yeah, so we think of at least in lab yeast, right? Like we think of lab yeast as growing in this kind of oxygen rich or like oxygen exposed environment, which is kind of comfortable, and cells divide and they're happy. They don't have to do this anaerobic respiration. But in the brewery, that's not the case, right? We're kind of evolving or like selecting strains that do well in these fermentative environments. And obviously, there's reasons we select those strains is because over time, these strains have evolved to kind of deal with these stresses better than others. Doesn't mean that they don't feel the stress. It just means that they're doing a better job of handling it. So, for example, like too much alcohol is bad, right? It's bad for everyone, not just for humans, but also the yeast that make it,
Yeah.
because um, you make a lot of it, and what ends up happening is it starts messing with the cell wall and the membrane fluidity of how the yeast cell behaves. So obviously, too much alcohol can cause you to break the yeast cell and then lyse, and then they die. And then things like sugars, again, like too much sugar is bad for humans, too much sugar is bad for yeast too, right? You can't have, because they have this osmotic stress or any kind of nutritional stress where they don't know how to respond, because the natural tendency is to take the sugar and break it down. But if you just keep giving sugar over and over again, then of course it can lead to cases where it doesn't become beneficial to the strain anymore. And I mean, I think more than the sugar, I think the alcohol is like a big stress, but more than that, it's the starvation from oxygen, right? Like as yeast strains ferment over time, the amount of oxygen in the tank keeps going down. And obviously, yeast likes all this rich media that contain all the amino acids, proteins, sugars, and carbohydrates. But when you start depleting the oxygen, the yeast really starts feeling the stress because you're shifting its respiration, which is like central glucose metabolism, which is central to almost a lot of the pathways in yeast, is getting shifted away to make ethanol. So it really needs to adapt. So we really were curious of like, hey, can we actually take an industrial fermentation environment and then look at these changes? Because if you look at it, then you can understand what's going on.
Yeah, yeah. You look at it, you can understand what's going on. Because like you said, this shift from respiration to fermentation, I mean, yeast cells are capable of doing both. They're capable of respiring and fermenting, which is cool. So like, how does the cell know when to change? And that's protein related, right? That's like osmotic stress related. It's reaction, to different stressors. It's presence of sugar. It's ethanol. All of these things impact what the yeast is actually doing at a specific time, right? As the ethanol increases, you need more resistance to ethanol. So you're going to be doing things that affect your cell wall. But at the beginning, you might not need as much resistance to ethanol. You need more resistance to sugar, so the cell might be reacting differently. I think of it like It's like the skin on your body. When you go swimming or something like that, you're in the water, and if you stay in the water for too long, your skin gets all wrinkly, right? And then if you nick yourself, like on the bottom of the pool or on a piece of coral or something like that, you bleed because your skin is sort of, not as tough as it would be if you were just dry on land. Same thing if you're on land and you're out in the sun and you've got this big sunburn, your skin starts to burn, and so you need to do something to cover it, right, or to fix it. And so the yeast has these same sorts of stresses. It's just inside of this anaerobic environment, which is cool. And that also reminds me of one other thing that we talked about with Lance Shaner, who's from Omega Yeast. Talked about this several episodes back, where we were looking at. I did an experiment looking at olive oil and how olive oil has some fatty acids that the yeast needs in its cell wall, but it doesn't have sterols. It doesn't have ergosterol, which is a big component of the cell wall. We talked a lot about that in that episode. And so that's an example of something that the yeast is having to produce proteins to help synthesize, right? It needs to synthesize ergosterol for its cell wall. So the yeast is doing a protein to that. So one of the questions that I have to ask is, like, how does the yeast manage to adapt to all of these different stressors? What does the yeast do?
Oh yeah, so taking your analogy of diving in the water a little further, so you can imagine if a regular human being is like a wild yeast that you get in the lab, you're gonna face the stress harder than if you're a yeast that's fermenting. And so you can think of yeast fermenting as like a free diver or a scuba diver, where the skin is like more adapted. They can hold their breath in the water for longer, so on and so forth, right?
Yeah. That's a great example.
But yeah, I mean, the stressors, how they do adapt. It's really interesting. You guys bring up the ergosterol story, because we do see some of the ergosterol pathway is one of the clear signals that we see in our data, where it's like over time it's adapting to turn on ergosterol synthesis. So it turns out, I mean, this has been the power of working with baker's yeast and brewer's yeast is there's like a 30-year plus catalog of research and just a whole body of work, which I think it's up there in terms of the most profiled organism on the planet, especially Saccharomyces cerevisiae. And so we can look back and compare to all these different literatures and see exactly the same thing. It's like, so ergosterol is a stress response, and it's been well documented, especially when you start limiting oxygen and increasing alcohol production. So, and the reason for that is like the yeast is trying to make more of its membrane lipids so that it protects the cell. And so that you don't struggle with osmotic stress, you don't like have issues in terms of, like survival, and you become more resistant. So ergosterol response is like one of the most standard things that you see in a stressed yeast population. And it's very gratifyingly for us, where we're able to see that pathway, really get turned on later in fermentation, and especially longer during serial repitching.
Yeah. And it is. So it's like the yeast is actually producing, it's changing itself to, or not changing itself, but it's activating or different genes become active to create things or to help them with stress responses. And so that's pretty important. And I wanted to ask a question about, you mentioned that like yeast again is well studied. There's a 30-year history from it. And yeast actually was, I think you mentioned this in the paper as well. It was actually the first microorganism to have its genome fully sequenced. And so I wanted to talk for just a second about what that means to have your genes sequenced, and then also the concept of expression, because just having the gene present doesn't necessarily mean that the gene is active or that it's doing anything. So talk to us about those two things. I think that'll help us understand the results a little bit later.
Yeah, no, this is a really great question that you raise, because just like us humans, we have like 3 billion odd letters or strings of DNA, letters in DNA that make up the human genome, which we now know from the Human Genome Project. Actually, yeast was the first eukaryotic organism to be sequenced. So before they did the human genome, we already had the yeast genome. And so unlike humans, lab yeast strains are like much smaller, and they have on the order of 12 million letters as opposed to 3 billion letters. So sequencing is literally going and reading the string of letters of DNA that an organism has, right? So that's like So you go start from finish, you start at one chromosome, go all the way, and then read through one chromosome, and then read through all the other chromosomes. So now you have a genetic map or genetic sequence, and then the aggregation of all that is what the genome is. And so that's the genome. But expression is a very important point because knowing whether a gene is present or absent is what sequencing tells you. So it tells you the genes or pieces of DNA that are present and absent. But what we don't know is how those genes are getting turned on and turned off. At which states do they get turned on and off? And that's where expression comes in. So the genome is like I said, is the recipe book for the cells to make the recipes, which are ultimately the proteins. And these recipes are transcribed into an intermediate step, which is called RNA or mRNA, which then ultimately get translated into the protein. So proteins are the functional molecules of cells, like I said, and they perform all these biological functions. So it's hard to know which genes are expressed or turned on or turned off just from the DNA sequence alone. You can know what's there and what's not there, but you don't know how they're turning off and turning on. So by measuring RNA or proteins, we can now infer which genes were expressed. So the higher the gene is, like the more it's expressed, right? So if we measure that at the protein level, you know that that gene is required at this time, so that's why the cell is making more of it. But you get that dynamics only when you read the RNA or you read the proteins. So that's the difference between expression and just sequencing.
Yeah, it's
given that RNA is also sequencing, but you're measuring the counts or you're counting the number of RNA molecules that are there. Same thing with protein. You're kind of estimating the number of protein molecules or the amount of that protein that's getting expressed at a particular time.
Yeah, so it's like measuring the amount of proteins that are produced, and that's telling you, okay, this is what the yeast needs at this point. This is the amount. This is what the yeast is focusing on right now, because we can see this is the protein. These are the proteins that are being expressed, and this protein does this function. So we know that the yeast needs or is doing this function right now. This is essentially what it's doing. It's expressing this protein that the enzyme that reduces acetaldehyde to ethanol. Okay, yeah, it's great. It's also expressing this protein that's increasing ergosterol, right? And these sorts of things. And this is where we can start to really understand the functional aspect of the cell. And so to your statement earlier, like about sequencing, I think of like DNA as like, it's like the library of instruction manuals, right? It's like this is the instructions for what you need, and here's the library. And then, but you don't always need everything at one time, right? You go to the library and say, hey, we got this problem with ethanol, and go to the library and the DNA says, okay, build this protein, right? And then that gets taken out by mRNA, and then it gets translated into the protein, and the protein actually creates an enzyme or whatever, does whatever function the cell needs. So the genome is just like the storage area. It's just the information data bank. And then the proteins are the ones that are actually out there doing it. Those are the people that are out there constructing the house, right? Or building the house.
Yeah. So your floor plans and your blueprint is what the genome is. And then the way you use it and what gets made into the final house is like they're the executors. Right?
Yeah,
so that's how I see it.
And again, you make the point that this is different, right? In every cell, in every different type of yeast, they have different blueprints, and thus they make different things. And so one way we can look at this is we can look at the blueprints and see what information is even available, right, in the cell. But we can also, and maybe I'm not going to say more importantly, but also, we can also look at what is actually made, right? Like what is the cell doing and what are actually being expressed. Yeah. And that's the proteomics aspect.
Yeah, absolutely. The genome project is amazing, right? Like DNA sequencing is like one of the most transformational tools that technologies that have been developed over the last, decades or two, and really helped us map all these genomes and tell us like, hey, these genes were there and these genes were not there. But now we're kind of starting to get at, well, okay, now we know that these are there, but like now we know what they're doing, right? So we're looking at it across time, across conditions, so on. So I think there's, it's not, I don't think there's one or the other technology. We need all of it because we're asking different questions with each of it. So
yeah,
they're
you're
all
asking.
complementary.
That's totally right. You're asking different questions with each of it. You need to know what the blueprints are there, because if the information doesn't exist, that's important information, right? I think of like another, perhaps a really easy brewing example is phenolic off flavor, a pop positive yeast or a pop negative yeast, or hey, about the one with that Laura Burns was just talking about on Omega yeast, the hazy one, right? Like some yeast have higher expression of this hazy one yeast or hazy one gene, and other yeast don't, right? So some yeast have a different amount or different information. Their library is different than others. That's important to know. It's also important to know which things that yeast is actually making. And this kind of brings me to one huge component of your study that I don't want to overshadow. And we mentioned it a little bit earlier, but this work will help us understand how yeast evolved and will evolve in the future. That's because you're looking at how, what genomic changes and what proteomic changes are occurring during fermentation. And if we understand how yeast evolves, we can use this learning outside of brewing, like health and medicine and all that. So talk to me about that aspect of the study, like use outside of brewing.
Yeah, so I think there's 2 points I would like to touch on. Like the use case of the power of doing these large scale studies in especially organisms like yeast, because one is that they are genetically tractable, that you can do these studies, right? But then the power is that now you can relate to, yeast and humans still after 1 billion years of evolution share a lot of the genes, that they all came from the same ancestor. So you can imagine thinking of, and the fermentation environment is a very controlled environment. So you can go in and tease out the genetic factors or the chemical factors and look at the stress responses of yeast genes, and then like compare them and see how many human genes are the same, behave in the same way, or they go through the same trajectories, or they respond in which pathways respond in the same way. And look at, you can also get at core underlying biology of how these pathways have evolved and like how they respond to stresses. So like that's a very obvious example of like, looking at, and this is the modern foundation for most biotech and pharmaceutical companies, is that they use these studies in model organisms like yeast and the worm and fruit fly and so on. And this whole vast knowledge about human health has been built up from these model organisms. So the more you sample new conditions in model organisms, the more likely you are to reveal valuable insights into human health.
Yeah. And again, that's a huge aspect of this study, right? I mean, that this stuff is, if we're developing fundamental knowledge of model organisms, we can use that fundamental knowledge in things other than brewing. But of course, we are interested in what happens during brewing, right? This being a brewing science podcast. And so, Riddhiman's study looked at how the yeast changes during fermentation, and then how looked at how the proteins that yeast makes change during fermentation, and then how those proteins also change after serially repitching. So we'll get to those results right after this break. One of the biggest improvements to my brewing practices was the upgrade to stainless steel, and Delta Brewing Systems offers some of the lowest prices on high-quality stainless gear, like the Ferm Tank, which holds 8 gallons or 30 liters of wort, comes with a domed lid to reduce the chances of a messy blow-off, and it can hold up to 4 psi of pressure. Delta Brewing Systems also has their own line of brew kettles, as well as one of the lowest-priced all-in-one electric brew systems out there, and their prices are remarkably affordable. If you're in the market for legit stainless gear that won't break the bank, you've got to check out deltabrewingsystems.com. We all know that designing recipes is really fun, and doing it well is so much easier with good software. We at Brewlosophy recently made the switch to Brewfather, and honestly, y'all, we could not be happier. Brewfather utilizes the latest technology to bring you the most robust cloud-based recipe design software that can be accessed anywhere, on your phone, tablet, desktop, and even offline. It also works seamlessly with numerous third-party devices to make it easier to monitor every step of your brew. I know change can be difficult, but trust me when I say you need to go to brewfather.app today to try it out for yourself. That's brewfather.app. Yeast is doing a lot during fermentation, and the functional aspect of those changes are happening through proteins. Now, as part of this research, Riddhiman, you profiled lots of different proteins. Do you have a favorite brewing yeast protein?
Oh wow, it's a hard question. Probably stumped. I don't think I do actually. It's um, for all this technology that I'm talking about using, I'm a Luddite in some ways that I still collect vinyl, and it's like asking me to go into my vinyl collection and pick my favorite record. It's kind of hard.
Right.
Everything is everything is interesting in its own ways, and it depends on the kinds of questions that we're asking, where we kind of focus on subsets of the proteome. So
Mhm.
I'm really unbiased. I wish I was more biased, but I'm unbiased.
Well, good answer. I'm sure the proteins will appreciate that. And of course, the proteins do all have functions that are important. Otherwise, the yeast wouldn't be doing it. I mean, that's also a way that evolution happens, is like jettisoning parts of the genome or parts of, proteins that are no longer functional. So that's also a piece of this. This whole, discussion of how yeast changes during fermentation and after repitching is also sort of a discussion of evolution. So I like that answer a lot. Well, I want to get into the methods of your studies, like sort of the study design, so we can talk about what you did and then get into the research about what proteins you saw change over time. So why don't you walk us through sort of what what you did, like what yeast did you study, how did you do it, and all that.
Yeah, cool. So we started. We started with the Hefeweizen yeast strain, which is the Weihenstephan yeast clade. It's a Cerevisiae strain. So yeah, there's we went after Cerevisiae strains because of like three or four reasons. The first one is that, like I mentioned before, like it's Saccharomyces cerevisiae is among the most profiled organisms on the planet. So you have this vast body of literature that you can go back and reference and look at and compare our data with what's already out there. The other one is that the Hefeweizen brewing cycles are pretty short. So, it's like typically 4 to 6 days. So it's easy to kind of do things in a short time span to look at things, especially when you're trying to wrap up your PhD and graduate and things like that. And
Yeah, I feel that.
and then I think the third thing was because they were really curious because of the POFs and things like that. There was some motivation to look at phenolic off flavors and see whether we can detect any enzymatic changes in the proteomics, and we're interested by that. And also, I think having the one was a very practical reason, was because like you guys know that ale strains are top fermenters and lager strains are bottom fermenters. Like when we are sampling yeast during fermentation, we are sampling them from the tank through the spout, right? So, but if you sample a lager strain, it's likely that all of them are at the bottom of the tank, and we actually don't get much yeast out from the top. So we picked this for a very practical reason. It turned out it was a good choice because we're seeing all these fun stuff going on. So and it turned out that we collect plenty of yeast even in this setting. So that's one of the reasons why we did that.
And then of course
and then,
another very practical reason too is like Live Oak Brewing being a partner, right? Like why that Live Oak Hef, the Weihenstephan Weizen yeast that they're using for the Hef is that's a bread and butter for them. So they're really interested in knowing what happens with that yeast too. And Live Oak is not the only brewery in the country using that specific strain to make Hefeweizens.
That's true. I mean, to be honest, Chip is so gracious that he would have probably let us profile anything we wanted. But
Yeah.
but it was, no, we were excited. I mean, the fifth most important reason, I think, is that because we loved the Hefeweizen beer so much that we were just like, we got to profile this.
Exactly. Well, and of course, as we always say on this, all the research just leads to more questions and more research, right? So you've done this now with the Weihenstephan yeast. It's only a natural extension to continue doing it with other different yeasts as well. So again, you used the Weihenstephan Weizen yeast. That was the yeast that you picked to look at. So then you sampled this, from the top of the tank and then ran it through some proteomic analysis. So talk to us first about like the number of times and when you sampled, because that's an important part of it. And then let's talk about the proteomic analysis.
Yeah. So No, this is a really great question because that's a question we had too. It's like how often do we need to sample and how many samples do we need to collect? And firstly, whether we would get enough samples from when the yeast are pitched, right? So um, yeah, so we started we wanted to do it across serial repitching, so there's going to be at least two repitchings. And then what we did was within a particular cycle, we sampled, I think, uh, depending on how you count it, it's four to six. Totally across the whole experiment, we did 17 time points, and then those were split up by 7 time points for the first repitch, and then the 7 time points for the second repitch, and then there were 3 time points from like a conditioning tank where they just let the yeast sit for a while. So we picked the conditioning tank just as an outlier to kind of see whether, because here the yeast strain should be doing something different, uh, and we do see that that's often the case. And then we did match time points. So if we pick the time, mostly match time points, apart from 1 time point in those 7 time points in yeast, where we sampled right at the start of fermentation through the end. And that was like the first day we sample the, either 3 hours or 6 hours after they start the fermentation, through 1 day, 2 days, 3 days, 4 days, and then finally after they crash. So that gives you 7 time points. And so we thought this would be a good starting point where, this gives us the whole range of fermentation, and it also gives us the repitching and the conditioning. So
Yeah.
that's what we did there.
Yeah. So again, like it's looking at sort of two aspects of this. It's looking at what happens during fermentation, right? During that time window in a single batch. And so for ease of use, I think we'll refer to it like you did in the paper. And we'll call that first batch 1. That's a fresh pitch of yeast straight from the manufacturer going straight into a fermentation tank, batch 1. And then you did 7 time points of that yeast. So right immediately as you pitch it, and then, while fermentation is ongoing, and then at the end of fermentation as well, pulling it out. And then the live oak repitched that yeast in 14 different cycles of beer. And then on batch 15, which is what we'll talk about, batch 15 is where you did the harvesting again on those same time points throughout that fermentation. And then a keynote here too, like the conditioning tank is a really interesting part of this as well, because a lot of brewers are using a conditioning tank, especially either pre-filtering or post-filtering. It's a place where beer goes and sits cold under pressure so that it can carbonate and it can yeast and sediment and all that stuff can settle out. So yeah, that's interesting to see what yeast is doing in that situation as well. Mm-hmm.
Yeah. I mean, yeah, exactly. That's one of the main reasons why we did the conditioning tank, is like we expected that it's the environment is changed from when it was fermenting, right? You're just allowing the yeast to sit, and so carbonation and things like that. So we do see certain subsets of proteins that are only specific to the conditioning tank.
That are specific to the conditioning tank, which is interesting too, right? And then that sort of makes sense. And I want to get into that whenever we get there. I don't want to forget about those to talk about the conditioning tank too. It's easy to focus on the fermentation because that's where the bulk of the data is, and a lot of the really interesting stuff happens. But the conditioning does too. And then I wanted one other thing too that I think is really important to talk about is the yeast management, right? So how the yeast was serially repitched, right? Because you are profiling Saccharomyces cerevisiae and that same yeast, right? That's changed over the 14 cycles. So so the yeast was pitched directly from batch to batch. They weren't growing the yeast or introducing any other things. It was just going batch, like most brewers do. And again, the yeast was harvested at roughly the same time points for each batch, and samples were taken directly from the fermentation tank. So not pulled off or, sampled somewhere else or anything, like trying to make this as pure as possible, that we're just getting the Saccharomyces cerevisiae and only looking at those changes.
Mm-hmm. Yeah. So that's exactly right. I mean, the yeast management was, if you mean like how we handled it, this is an important point because brewing strains are coming off of this kind of anaerobic environment, and the moment we sample them, they're exposed to air. So we need to make sure that, and some of these proteomic changes are very dynamic, right? So if you let them get exposed to air for too long, then they might change on the way, the time between when you take them to process to to what they were behaving in the tank. So this was a key concern. And so what we did was we would sample, freeze the cells down so that they are freezing even their dynamic state at that point, and then taking them to the lab and then processing them after. So, yeah, it's something that if people in the lab often we take for granted. Like there are a few specific cases where you kind of focus on, but when you pull something that is heavily adapted to an anaerobic environment and suddenly expose it to air, there's going to be all kinds of
Yeah.
the yeast is going to be like, oh, what is this new thing? And I'm going to like, I thought I don't have this thing, but now I have this thing. And so we need to like freeze them to be like, no, we want to sample how they were like in the fermentation tank. And so that's what we did in the yeast management. So we collected samples. It's literally me and another colleague of mine who would like drive down to the brewery, collect samples, freeze them, drive them back to the lab, process them, and store them in like deep dark cold storage so that the cells are kind of stalled in terms of dividing or doing anything. And then after that, we would process them all together to extract the proteins and then do the analysis.
Yeah, right. And that's the key point that I wanted to make, right? We're making sure that we're not introducing any sort of other factors by our sampling method or by yeast management or by repitching or any of those things. We're trying to be as true to what's happening in the fermentation tank as possible. Now, you used, again, we've talked about it several times on this, like proteomic analysis, and you use something called high throughput shotgun mass spectrometry. And this is a pretty, as I understand it, this is a pretty modern tool that helps us understand how proteins change or how the proteomic, here, instead of me stumbling and looking for words, how about I just ask you, what are some modern tools that are available and how do we study protein expression changes in yeast?
Yeah, so no, this is great. I mean, question, right? Because the last couple of decades, our ability to go from measuring just one gene at a time to hundreds and thousands of genes at once in like this high throughput fashion has really transformed biology. Right? So previously, before, one of the ways that people did proteomics was, like you can imagine purifying or isolating a particular protein of interest, like for example, alcohol dehydrogenase that like makes alcohol, ethanol basically, and you can take those enzymes that produce these alcohols and studies how those change. So that would be specifically isolating only that particular protein, right? So, but obviously that is not scalable because yeast have something on the order of 6,000 odd proteins, and so doing that for all 6,000 is a long side project or even like a long career, right?
A long
Some ways,
career.
but,
Yeah, for sure.
So scaling and studying to many genes becomes very difficult. When that's where like omics or like high throughput technologies come into play, where we can go from it's not just for proteins, but DNA sequencing has the same thing, RNA sequencing has the same thing, metabolites too, where technologies have been developed now, where instead of looking at one gene at a time or one protein at a time, you can look at a lot of proteins or hundreds or thousands of proteins, right? So, and then this is really important now because proteins, as we know, rarely work in isolation, but they often work in concert with each other to form these modules and metabolic pathways. And it's hard to know these interactions and how these happen and their consequences for the yeast cell without studying all of them, at the same time. And today we can ask those questions at scales which we couldn't before. And so, in our case, we sampled close to like half of those 6,000 proteins to gain this global view and not looking at only a single view, but entire pathways and how entire pathways and protein modules shift over time. And so, yeah, I mean, so if you talk about shotgun mass spec, basically what it does is you lyse the yeast cells or you break them open, and then you isolate the proteins from the cells. So get rid of the DNA and the RNA and get and the sugars and all of the dead yeast and then all of that. So you have the proteins that you have isolated, and then what you can do is you use enzymes called proteases, which I'm sure people are aware of, where you can use that to digest the proteins into smaller pieces called peptides. So you take an entire protein, break it up into small pieces, and then what you do is then you take those peptides and then you pass them to a spectrometer, which basically separates proteins based on their mass to a charge. This is obviously oversimplified, but essentially you get an abundance measure, or how much of the protein is present in that particular sample, right? And so you can do that. That's just for one sample, and then you do that across multiple samples, and then what you get is this kind of measurement of that same protein across time, across all these samples. And so then you're actually measuring how these proteins change over time. And so mass spec allows you to kind of parallelize that, or like mass spectrometry with especially shotgun mass spectrometry allows you to uh, parallelize that in a fashion where you can really deeply sample the yeast proteome. So something like we sampled close to 5,000, but we can if there's ways to go higher than that. Not 5,000, sorry, 50% of the proteome, close to that, but there's ways to go higher than that. So that's how it works. So if you take a cell, break it, isolate the proteins, break the proteins into small peptides, shoot them in the mass spectrometer, and then those will give you a mass to charge. Every peptide has its own fingerprint of mass to charge ratios, and then from that you can compute the abundance. And so you get an abundance for a single protein, and then you get that across all the proteins that you saw, and you see how those change across time.
Yeah, on each sample, right? So you get the amount of proteins that are present in each sample, and then you can apply that and see how it changes over time. So again, I just want to give sort of an example of the amount of work that this was done. So you actually did 64,740 protein abundance measurements. I mean, like, that's, if you were isolating those individual proteins and trying to figure out the abundance of those proteins, that would take decades to do.
No, I mean, it's like, thanks to, high throughput technologies have really kind of pushed us forward, right? And
right.
And yeah, I mean, so that is especially in in academic science and research, this might be what's considered like high throughput, but it's on the low end of the high throughbut. But
Sure.
But for brewing science, it's like really pushing the scale forward, because we were looking up this while we were studying, while doing this work, and I think the previous study sampled something on the order of, if I'm remembering correctly, something like 20% less than what we sampled, if I'm not wrong. And so, no, sorry, I should correct myself. It's 133 proteins is what they saw. And given that was a while ago, but we're seeing 2,500. So it's already a big jump, right? So
yeah, a huge jump, right? Looking at 25 or 2,600 different proteins, and that really allows you, like you mentioned, using this technology allows you to take this sort of global perspective of what happens across the fermentation and then across cereal repitching. So from this global perspective, if you're just looking at kind of like all the proteins change and looking at the abundance of proteins across time, what did you find? What did you see?
Oh yeah, I mean, it's really cool because we see known things and then we're seeing like new things. And one thing that was very exciting for me was the power of taking this like systems biology approach, where you can look at multiple things at once. In our case, it's 2,600, but across many time points. What we can see is that like we're looking at the proteome getting regulated over fermentations, right? So what's interesting is now you can take every profile of a protein across time and then start clustering that data. So which means that you can start grouping proteins that behave similarly or change similarly in the fermentation tank. And once we did that, immediately like obvious patterns started falling out, which is clear in the data. One was that the proteins that form the ribosome. So the ribosome is this massive protein complex whose job is to make more proteins. And by reading or translating the mRNA, which contains the recipe to make that particular protein, we see that the cells that make up a lot of these ribosomal proteins and this machinery, which we call the translational machinery, goes up in the first 6 hours of fermentation, and then like drops down after that. And that's not only in one batch, but we see it across both batches, and it's very reproducible. So, and the signal is very obvious. So from 0 to 6 hours, you make the things that you make the machinery that you need to make the other proteins of the cell. You turn that off, and then you see the rest of the metabolism, like sugar metabolism and fat metabolism, turn on. So like that's just one example I'm talking to you about, but it's the most clear example that we saw, and that's just very satisfying to see these like systemic shifts which we would not be able to get at using, like a one gene or one protein approach.
Yeah,
Right.
totally, right? I mean, you're able to see like what is the yeast doing? It's like here's the product. Here's what the yeast is producing right now. This is what it's doing at this time. So in the first 6 hours, we talk about that. Oh, I guess we didn't talk about that in the last episode. Maybe we can. Maybe I can gloss over it for now, but if and then Jordan and I can touch on it next week. But this this concept of lag phase, growth phase, and then maturation or death phase, right? So as I understand it, lag phase is kind of like where the yeast is building up, like you said, the machinery that it needs in order to do things, in order to make new cells or grow or do all that stuff. And then once it's got the machinery, it can enter growth phase where it's budding and where it's reproducing and growing and creating new cells, and those new cells are then going on and creating new cells. And so that's the exponential growth phase, which we see. And then there's this sort of hibernation or death phase, right, where the yeast is sort of shutting down. It's done everything it can do with whatever information is in its environment, and then it's shutting down. So do you see something like that when you look at the global proteomics?
Yeah, I think so. I mean, I think that's very that's basically what we see, because there's the added pressure of yeast has to do this growth in like a fermentation environment. So it's not quite exactly the same rate kinetics at which the yeast take off. So the exponential phase is probably more delayed, and the slope on that is like much lower, so it takes longer. But what we do see is exactly that. So in the lag phase, you're building up all these we're building up the ribosome, because the ribosome needs to make all the other proteins in the cell, and then later you start seeing things like sugar metabolism and fat metabolism. And then like once you start reaching the lag and stationary phase, that's when you've like depleted a lot of sugars in the wort, and you're struggling to kind of survive. So your population really stops dividing as much. So, and that's kind of reflected in them shutting down a lot of their protein machinery just to kind of make sure they can stay, because making a protein is not cheap for a cell, right? It's pretty costly. You got to make the ribosome first, and you got to translate the mRNA, and you got to transcribe the mRNA. So it's like you have to adapt to kind of being efficient. And also, we're forcing the yeast to make ethanol for us, right? So there's also that added pressure. So
right, yeah, we wanted to make ethanol, but that's an important part of this too, right? Proteins are made up of amino acids, and those amino acids have to come from someplace. So, like I was just talking about with Clem Fox a few weeks back, free amino nitrogen (FAN) is a measurement that brewers are interested in, because we want to make sure that there's a bunch of amino acid building blocks that yeast can use to make proteins. Like you said, it's costly. If they also have to make the amino acids, then they got to make the machinery to do that too, right? So they have to, if there are amino acids that are missing from their surroundings, the yeast can create some of them, and so they have to build that machinery. And that was, to me, there were two things that stuck out to me from the paper, which you talked about in like the batch 1 fermentation, especially at the beginning. You saw a high abundance of proteins that were involved in aerobic respiration, so like oxygen respiration, not fermentation, and then this amino acid biosynthesis. That was pretty cool to me to see, like, wait a minute, we're doing fermentation, but no, the yeast is actually at the very beginning of fermentation doing a lot of, or maybe potentially doing a lot of aerobic respiration.
Yeah, no, this is kind of I mean, it's good to see, but we expected to kind of see some of that, because when the yeast is when I guess the slurry that gets loaded into the fermentation tank, right, it's exposed to oxygen. It's not like expecting to go into the fermentation tank being under fermentation conditions, right? So it's actually gratifying for us, because it tells us that there are these characteristic shifts where it used to have aerobic respiration on, but as it goes into the tank, it shuts it off, because now it needs to turn on all the anaerobic machinery. And so it's like really kind of gratifying to see that. And also, like, now you can gain these new insights, because now you can start thinking of, well, are certain genes that are supposed to be on not being on or turned off. And so now that this gives you like a catalog book or an encyclopedia of genes to kind of go through and look and see, oh well, my gene is doing this at this time and is doing this across this time point, and so why is that, and like why is it doing that, and how is it responding or communicating with other genes in its same pathway or same process and so on.
Yeah, yeah, and again, like looking at this as it's pitching over, or, serial repitching. You have this aspect of, like you said, in the first pitch, the yeast is exposed to oxygen because it's that's probably how you grew it up in wort that was exposed to oxygen. And then when you pitched it into the fermentation tank, it's got some aerobic respiration, and then it's got to shift over to fermentation. But the serial repitched yeast is exposed to anaerobic conditions, and it's getting repitched under more or less anaerobic conditions, or at least it's primed that way. So did you see the aerobic respiration stuff happening in batch 15?
No, actually, we did see that aerobic like if you take the starting populations of both batch 1 and batch 15, basically, if you take that and then you look at where the same starting populations, but then you see that there's a difference in even in those populations where like the batch 1 and the batch 15 have changed, where the batch 15 has tended to adapt over time to now start actually being very specific to knowing that it's going to be in the conditioning tank for or like knowing it's going to be in this fermentation environment for longer, that you're seeing that like it's actually turning on those stress responses like like alcohol metabolic processes, like ergosterol processes, whereas in the batch one's time point, the starting time point, those are not necessarily on, right? So those have all the amino acid biosynthesis and ATP synthesis and cellular respiration being on. But when you go to that same time point in batch 15, it's actually off. And then the stress response turns on, like the sterol metabolism and so on. So it's like really clear, right? So now you're seeing that even though you're putting them in the same conditions, over time, after having repitched them, the yeast has adapted to know that it's going to be in this, or it's like being geared to be in this fermentation environment. And just seeing that at the protein level is really fascinating.
Yeah, like it's like the batch 15 yeast is like priming itself. It's like ready to go. It's like ready. It knows, all right, I've been in fermentation. I know I'm going back into fermentation or whatever. I mean, maybe there's not like, an actual cognitive thought there. But in any event, there's an evolutionary adaptation or some sort of adaptation that's occurring that those yeast are better primed to go straight into the anaerobic fermentation. Or, well, at least that they don't have to turn off all the aerobic machinery, right? I mean, who knows about the fermentation kinetics, but this is essentially saying that those yeast seem primed for anaerobic fermentation. They're ready to enter into an anaerobic environment, which is fascinating. And so, like you said in the paper, batch 15 is enriched for sterol biosynthesis. So that's that cell wall protein that we talked about, and glycogen and cellular alcohol metabolic processes. So that's, again, it's primed, ready to enter into fermentative processes. So I think that's really cool. And now you also mentioned two compounds in the paper that you kind of focused on here as well. One of them we've kind of already talked about, but there were some differences in like ergosterol and isobutyraldehyde. So let's talk about those two real quick.
Yeah, so the ergosterol was the obvious one where we saw that the whole pathway tended to be upregulated in the batch 15 versus the batch 1, just across, if you aggregate across all the different time points, we see that. But in the isobutyraldehyde metabolism, we actually see the opposite, where we see it's actually on in the batch 1 and then it gets turned off in batch 15. I think one of the biggest kind of genes or proteins that tended to get turned on was this gene called PDC5, and that's involved in this the decarboxylation of these alpha keto acids and convert them into fusylaldehyde. And then the fusylaldehydes get oxidized either to form fusyl acids or get reduced to form fusyl alcohols, right? So and now we're seeing that if you look at this Ehrlich pathway, this is the pathway that is involved in converting amino acids to fusyl alcohols. We see that that Ehrlich pathway is getting turned off in batch 15. And so that's the one thing. It's like these kinds of large-scale data sets give you very good starting points to now go and ask questions about why is the Ehrlich pathway behaving this way in this strain? How does the Ehrlich pathway impact flavor? And like how do these fusel alcohols change across serial repitching, and are they actually making it to the final beer product? And so that's like a really clear example. And so just as an example, I mean, these keto acids and fuselaldehydes and alcohols are all like flavor compounds, right? They have aroma, they have taste, they have flavor. So isovaleric acid, for example, is I didn't know that this was a known or a technically accepted term. It has this gym socks aroma, which
Yeah.
I didn't know about. Apparently, it's used as a standard to like train brewers, and butyraldehyde is responsible for that grainy flavor. And you're seeing that grainy flavor is getting like the pathway to make that grainy flavor is getting shut down. And so now this kind of raises the interesting question, right? So we made an observation, so now you can generate all these hypotheses to now go back and like see whether that's changing. One of the things that is very undersold in our study is the impact of the metabolites, that the compounds that are getting produced, because all these interact with the proteome too, because they feed back and tell the proteins that to stop making a particular compound in the pathway, right? And so we can go back now and follow up and say that, hey, we are expecting the Ehrlich pathway to change in this fashion. Now we can go back and look at the compounds that come out from the Ehrlich pathway and ask whether those are different across batches. And the hypothesis is that they should be, but if they're not, then there's regulation that's going on, which is even more interesting. So now you can start mining different aspects of the pathway in this way, and that's what this kind of a study gives you the ability to do, is now you're able to look at these things and ask these questions which we couldn't.
Holy moly. And that seems like an opportunity then for like even genetic engineering, right? Like for changing strains to better interact in these pathways, right? Or to change the way that the yeast interact or move down these pathways for different outcomes.
Absolutely. I mean, this is, I went to my first MBA meeting this past year. And I'm kind of sad that was my first one that I went to. I had so much fun. And one of the big takeaways I came back with from the conference was how fast yeast strain companies and engineers are moving towards genetic engineering their strains, and CRISPR is amazing. I've used it in the lab to engineer my own yeast strains and so on, but it's just amazing now what the possibilities are for engineering commercial yeast strains, in terms of first taking out flavors that you don't need, like POFs and things like that, right? But also what's more exciting is like people are actually using these gene editing techniques to introduce new flavors from plants. For example, like making a hoppy IPA without adding hops in it, right?
Yeah.
And so now this our resource is now offering candidate targets to now edit, change, improve, or even perturb, for example, genes or proteins, so that you can study them in fermentations and adapt these strains to now like perhaps even produce new beer styles that like once you can look at what's happening, you can think about how to break it, right?
Yeah, for sure. Oh, yeah, exactly. Or how to change it,
Or
how to break
change
it, how to
sorry.
change it.
Yeah.
Break it's fine too, right? Some things we need to break, right? But to use the two very specific examples from your paper, we talked about ergosterol, right? And how ergosterol helps the cell, to regulate these osmotic stresses. And we add oxygen in the brewery so that the yeast has the things that it needs to build ergosterol. But yeast over this, from batch 1 to batch 15, they had higher ergosterol. They had higher ability or better ability to produce ergosterol faster. I guess they had higher abundance of ergosterol type proteins that were there. So like the yeast was better primed after several batches to produce ergosterol, right? So then the first batches. So that's a very good, very specific example. If you could genetic engineer some yeast where you didn't have to use as much oxygenation, or the yeast were just ready to go because they had this machinery that's ready to go, that might be an opportunity for genetic engineering. And another thing, and maybe this one will ring in the ears of brewers more so than ergosterol, because that kind of sounds like, okay, that's a yeast thing, and I don't mind putting oxygen in the beer.
I mean, just to butt in a little bit, is that people have shown in the past that, you can supplement ergosterol into your wort and make the yeast like feel less stressed, because you can trick them into thinking that there's a lot of ergosterol around. So people have shown that oxygen stress response goes down by adding ergosterol. So it's just nice that we were able to kind of map the molecular level changes, right? But I agree with you, right? Like engineering the pathway to be turned on much earlier, or like stay turned on through fermentation, might be one way to go. And sorry, you were saying about the flavor component.
Yeah. No, I mean, that's a great example. And we did talk about that briefly on that episode with Lance Shaner about introducing ergosterol, or like making the yeast think that ergosterol is present so that it's not as stressed, because yeast stress will produce different flavors in the beer. That's a great example. So these are pathways that we can look at, but like I was saying, like brewers, might kind of roll their eyes at this and say, well, yeah, okay, that's a yeast metabolic pathway, and I don't mind oxygenating my wort, so it's fine. I not really something that I'd want to be interested in. But one of the reasons why a lot of brewers, there's a lot of anecdote out there I've heard from a lot of brewers that say that they don't like early fresh pitch batches of their beer. They like the flavors after 2 or 3 or 4 repitches, right? And that's when the beer really starts to grow. And this is an example, this isobutyraldehyde that you were talking about, that was less abundant in batch 15, you mentioned it's associated with grainy flavor. It's also lower production of fusel alcohols. So that grainy flavor goes away as the yeast is re-fermented, and there's lower fusel alcohols. There's actual flavor changes that you can see looking at this proteomic analysis. And so these are things that might be interesting to assess. Why is isobutyraldehyde less produced or lower in abundance in these later repitched batches?
Yeah, no, absolutely. I mean, and just tying it back to what we discussed before about POFs, and so like we know 4VG is like one of the key POF compounds that
4-vinyl glycol
that are present. Mm-hmm. Yeah. And so like if you look at FDC1, which is involved in that production, we can now map how it's changing across time. And it's interesting because in batch 1, it drops down in the early time points and then comes back. But in the batch 15, it continues to stay at the same rate throughout fermentation, right? And so, and like Hefeweizen strains, for example, are like really the characteristic clove and banana flavor, like, isoamyl acetate.
Absolutely.
Is the ester that does that.
4VG is the other one, right?
So the isoamyl acetate gives you that clovey banana flavor, and then ATF1 is the gene that's involved in that. And we were able to now map the dynamics of, in batch 1 that goes up and then goes back down, but in batch 15 it goes up and then doesn't drop as much down as what we thought it was, right? So now you can actually start exploring, because you can ask, well, if I knock out isoamyl acetate, the ATF1 enzyme with CRISPR, do I change the flavor of my Hefeweizen strain? So you have a candidate list now, right? So you can go down and look at, hey, these genes do this. We know that because of the past 30 years of work characterizing the yeast genome, basically. And then now you can like come up with hypotheses of like, I want to make a strain that has lesser clove flavor. So I'm going to turn this gene down. So I want my beers to taste less clove, or I want to make my beer Hefeweizen, which is super hoppy. So I just put in a bunch of genes from a different species and ask how this changes, right? So not that anyone needs a hoppy Hefeweizen in the world, but right. I'm just shooting ideas out there.
Sure. But certainly, modulating that clove and banana flavor is a huge piece of making a Hefeweizen, right? I mean, I'm sure this is something that Jordan and I are going to talk about next week is, how to make Hefeweizen, right? I mean, and use Hefeweizen yeast. So if you're able to change, and create strains that either up or down regulate these specific genes, you're starting to get information. And again, it's all because of this research that you're doing, right? This proteomic analysis is being able to say like, okay, we can look at the proteins, what functionally is happening in the yeast, and decide how those things are influencing not only just the yeast metabolic pathways, but also flavors of the beer, like what's actually being produced and what are you going to taste in the beer over time as the yeast itself is just serially repitched. I mean, again, we're not talking about, it'd be interesting to see like how different the strains are genetically. And I think I'll ask those questions of Dr. Dunham whenever she comes on. But it's interesting to see the proteins are different. And so they're producing a different beer. I mean, you might objectively have a different tasting beer from batch 1 and batch 15 if you paired them next to each other, if there's a way to do that. And so this whole analysis is fascinating and super interesting. And so I always ask this question before we wrap up the show, and it's if you want brewers to take away just one thing from today's episode, what would it be?
Oh, yeah, that's an interesting one. I mean, I just want to kind of champion or like be a mouthpiece to promote more omics type of studies in these industrial kind of settings, especially brewing, because craft brewing has been an art, but there's a way that we can combine it with science to kind of fine tune the art to go to places that people couldn't go to before, right? And so I want I guess like I want the listeners to take away the fact that like by making these global measurements, it actually yields very novel insights into the way we can like use this data to either engineer strains, optimize fermentation conditions, or even things like how to even maintain your beer in conditioning and so on, right? So I think that would be the one thing that I would say is that to take away is that like we need more of these studies, because our study is by no means like complete, right? So ours is a starting point, and we got pretty far in terms of getting at looking at all these different time points, but I think we're really scratching the tip of the iceberg. And so just imagine the possibilities of being able to now not only look at RNA, not only look at proteins, but couple that with RNA and the DNA, and also look at the compounds, right? So, and that's so I think there are people over at Escarpment Labs who I got to meet with who are like working like really hard on characterizing the metabolites, right? So on pairing that with yeast strains, what I think is going to be the next step, and I'm just excited about seeing where these types of assays can go in the future, particularly in brewing. And so that will be the one thing I would tell them to take away is that ours is just a starting point. It offers a resource to now ask your favorite questions, and we just hope that the brewers, we've made everything open access and available to everyone. So we want the brewers to take these resources and mine them better than we would ever do, and ask these questions in a more systematic and focused way than us, and use them for their own benefit, right? So.
Yeah. Oh man. Yeah. I don't think I could say that any better than myself. So I think I'm going to leave it there. Is there anything else that you wanted to talk about that we didn't get to today? I'm sure there's a bunch.
No. This has been great. I mean, I want to thank you firstly for this, for the invitation to be on here, and I've had a great time recording this with you. And I think the second thing I want to say is that there's a website that we've made for people to go and look up their favorite proteins and see how they change across time. And you can also compare your favorite 2 time points. So like, let's say I'm interested only in, batch 1, 0 hour and 6 hour. You can only do that comparison. So our website allows us to do that. And then the preprint is out, so you're welcome to read it and provide feedback. And so and I think that's it. And I'm sure you can feel free to reach out. I'm available on Twitter. I can share over my Twitter info, and I guess X is what I should call it right now. But
I guess, yeah.
But my social media is available for comment and discourse and even collaboration. So I'm just happy to be here, and that's it. Thank you again, Cade.
Yeah, no, that's awesome. And listeners, you'll have links to both of those. The website where you can check out your favorite proteins and see how they change over time. And then also the article as well, which, as he said, is open source. So it's free to everybody. Everybody can read it. Or not open source, but made available public. There we go. So Riddhiman, thank you so much for joining me in the Brew Lab.
My pleasure.
All right. Well, next week, Jordan and I will be back applying the science of this episode. We'll see y'all then. The Brew Lab is a production of Brewlosophy, where they who drink beer think beer. Don't forget to visit brewlosophy.com to read about our weekly experiments and other brewing adventures, and listen to us talk about it on our other show, the Brewlosophy Podcast. Thanks to all of our sponsors and patrons that help make this show possible. If you'd like to receive a reward for helping us do what we do, visit patreon.com/brewlosophy to see how you can do just that. Thanks so much for listening. We'll be back in the Brew Lab with another guest next week. Until then, think beer.